Mitochondria are football-shaped organelles that are roughly the size of a bacterial cell. They are surrounded by an outer mitochondrial membrane and an inner mitochondrial membrane. The space between the two membranes is the intermembrane space, and the space inside the inner membrane is the matrix space. The enzymes of the citric acid cycle, of the beta oxidation pathway for the breakdown of fatty acids, and for the degradation of amino acids are all found in the mitochondrial matrix space. The outer mitochondrial membrane has many small pores through which molecules less than 10,000 grams per mole can pass. Thus, the small molecules to be oxidized for the production of ATP can easily enter the mitochondrial intermembrane space. The inner membrane is highly folded to create a large surface area. The folded membranes are known as cristae. So the folds that are shown in here, these are the cristae. The inner mitochondrial membrane is almost completely impermeable to most substances. For this reason, it has many transport proteins to bring particular fuel molecules into the matrix space. Also, embedded within the inner mitochondrial membrane are the protein electron carriers of the electron transport system and ATP synthase. ATP synthase is a large complex of many proteins that catalyzes the synthesis of ATP. Not only are mitochondria roughly the size of bacteria, they have several other features that have led researchers to suspect they may once have been free-living bacteria that were captured by eukaryotic cells. They have their own genetic information, or DNA. They also make their own ribosomes that are very similar to those of bacteria. These ribosomes allow the mitochondria to synthesize some of their own proteins. Finally, mitochondria are actually self-replicating. They grow in size and divide to produce new mitochondria. All of these characteristics suggest that the mitochondria that produce the majority of the ATP for our cells evolved from bacteria captured perhaps as long as 150 million years ago.